So in our biology, class 11 topic, first you know that how we are going to proceed. That is very important. So one of the most important topic, what we find that we have already revised in case of class 11 also, that is plant physiology. That with which we are going to start with, that is plant physiology. Can you see the board?
Yes, sir.
Okay. Actually, since this is a that is online pro forma, so you need to interact. Okay.
Okay.
So
Okay.
plant physiology, first of all, you know that in our topic, that is plant physiology, photosynthesis is present, which I will be starting today. In higher plants. Then you have respiration in plants. And then growth and development. Plant growth and development. Then there is another topic which we know, that is popularly known as cell biology. We can divide our topic in this way. That is, first after doing this portion, that is a physiology, or whichever is your need, you can also tell me. This is one way I'm just designing the course so that it will be helpful for you to understand that this portion is done, then we can move on with another part. So biology. Apart from English, which language do you prefer? English?
But English, I prefer Hindi, Odia.
Okay, I know Hindi
Oh,
and Bengali also. Whichever, for the sake of understanding, I can talk in Hindi also. There is no issue. जब
oh.
कोई जरूरत होगा तो तुम मुझे बोलना, ठीक है? सेल बायोलॉजी में क्या हम करेंगे? पहले तो सेल द यूनिट ऑफ लाइफ which is there. सेल द यूनिट ऑफ लाइफ, देन उसके बाद है हमारा सेल डिवीजन, that is which is also known as the cell cycle. Division. Along with that, the portion which is present, this there is biomolecule. Now you tell me that this biomolecule section, is your zoology madam taking this, or shall I take this part? Do you have any idea that the cell biology include this unit of life, that is the cytology, which we also can call it as cytology, that is the study of cells and the chromosomes and also the different cellular structures which are involved. So that is called cytology. And cell division, it is also known as the cell cycle, where we will be dealing with how the mitosis, meiosis, the cell cycle regulation, and all these activities we will be studying, right? And after this cell biology, we come into the morphoanatomy. That means the structural organization, that is in case of plant, if we see that there will be plant morphology and anatomy. Often people are worried about this anatomy and also this portion, especially that is why I have selected the first topic. There are so many concepts are involved, many reactions are involved. So I found in my teaching career that many students are worried about that, what is this? And so morphology is not difficult, only the examples you have to remember. Otherwise it is easy, and anatomy is very good. That means it is, if you understand it, it is not at all difficult. Anatomy. After this, we are going to keep the classification at the last, because that is a part thoda sa ratta marne ki portion hai. That means you have to memorize those biological classifications. So I'm keeping all these good chapters before. Okay. Any
Okay.
any thing you want to say? from this?
No.
Then after that, which is the entire physiology section is coming, that is human physiology. I think ma'am will be dealing with that. And yes, there is no other portion, I think, that is after this morphology and anatomy in your NCRT. I don't think other part for class 11 is there. So entire physiology is coming under this portion, the cell biology, the unit of life, cytology, where genetics vagera will come in class 12 section, and morphology and anatomy. Just check your NCRT. Is any other portion regarding botany is there? Just check it out
Okay, so 1 minute.
in botany, because from there physiology, human physiology is starting. That is digestion to already nikal diya syllabus se. So you have breathing and exchange of gases that she will be doing, and then you have neural, endocrine, and I think one excretory, right? So
Yes.
only those portions are there that is she will be doing with it. So let us first start with our this part I'm rubbing. Let us first start with our this portion, that is the plant physiology. And in this plant physiology, I will be starting with photosynthesis. Redo করার কোনটা? Redo মানে suppose something has got rubbed up, so আমি কি আচ্ছা, okay, so I can, okay, then okay. So I can remove this page that is not whatever you have noted it down. So
Yes,
now
noted.
let us move. So the main source of energy. What is the main source of energy? The solar energy is the main source of energy.
Yes, sir.
And this energy can be trapped by group of living organism. Yes, कुछ group of living organism है. सारे के सारे living organism, all the living organism are not able to trap this energy. Those organisms which has the ability to trap this energy, they are known as autotrophs. They are known as autotrophs. Now, this autotrophs, from the name you can analyze, auto means self and trophs means synthesis. That means nutrition. Those organisms which can make their own nutrition with the help of solar energy, and they can convert them. They can convert this energy, one form of energy, to another form of energy, that is the food energy. You can also say, or you can simply call it as the chemical energy also. So, and this process of converting one form of energy into another form of energy, this is known as energy transformation. That means here you know that this is a solar energy, this is sun. So this one form of energy is getting converted into another form of energy. So this is known as energy transformation. Energy transformation, right? Because here you can see one form of energy, that is solar energy, has been converted into chemical energy. Up to here it's clear. So जब one form of energy दूसरे form of energy में convert हो रहे हैं, so हम इसको कहेंगे, what we will call it as
Energy transformation.
transformation. So now this energy which is stored in form of a molecule, they can be present whenever this energy is stored. They are stored in form of chemical bond. Whenever this bond they will break, there will be release or liberation of energy, which is known as minus delta G, right? Now this energy will be further utilized by a chemical moiety in the cell. You know that that's a domain which is present as a energy currency of the cell, known as the ADP. adenosine diphosphate. It will combine with inorganic phosphate, resulting in formation of ATP. So here from we can say that in this case, one form of energy, that is a chemical energy, is not converted into another form of energy. Rather, the compounds are different. Yes, the compounds are different. The energy form remains the same. So what we are going to call it as, this can be called to be as the energy transformation. Energy transformation. Sorry, transfer. TRAN is FER. That means when only the chemical energy is remaining as it is, only the molecules they are changing. When it is an energy rich molecule, it will break. This happens during the breakdown process. That is catabolic process. This is a catabolic process because there is breakdown of bond. Now here the bond, they are breaking out, they are breaking down, and the energy is getting liberated. So we can say that this is a catabolic process. But we know that in the process which we are going to start with, the things are going to get synthesized. Why? Because you know that autotropes is involved, and autotropes are responsible for conversion of this one form of energy to chemical energy. That means this chemical is going to get synthesized inside the plant. Yes or no?
Yes, sir.
So here we are going to say that this process is anabolic process. That means one form of energy, it is getting converted into another form of energy, and that is stored or retained within the molecule. So that will result in synthesis, that is anabolism. That will result in synthesis, and that is known as anabolism. Now in this process where we see that this energy conversion is taking place, this process is known as photosynthesis. We can take it up with that. This is where one form of energy is getting converted into another form of energy by a group of organisms that is known as autotrophs, and they are going to retain them, they are going to store them in form of chemical battery, which is known as ATP, right? So why we are saying this, that is this, if we analyze the word, we are coming out with the two words. One is photo, with the help of, that is light, and synthesis, that means manufacture, or you can call it as genesis. Synthesis का मतलब है to synthesize, that is manufacture. So what is being manufactured, or you can say to put together, to put together. Now what is being put together? There are a number of components, or you can say the raw materials which are involved in photosynthesis, which you have studied in junior classes also. So those materials are being put together and mixed up in a definite proportion, resulting in formation of what is known as the chemical energy or the food energy, right?
Yes.
If we see, if you can recall that what are the substances which are required as a reactant in the process of photosynthesis. Can you tell me? What are the substances?
So
Yes.
substance that is green pigment known as chlorophyll, and
Chlorophyll.
then
Apart from that,
sunlight, CO2,
yes,
water.
carbon dioxide is a necessary substance. Along with, there is requirement of, first I'm listing what are the things which are required, then we will come to the definition, what is photosynthesis, and then moving on. So carbon dioxide, light, it is required. Anything
Yes.
else?
Water.
Water. I'm writing H2O.
Okay.
So these are the things, and along with that, these are the external factors. Along with that, many other external factors, that is internal factors, are involved. internal factor is the chlorophyll. Now, there are a number of scientists. Now, this we know today, right? Today we know all these chemical substances, these raw materials, they are very essential for this process. But when this process was not discovered and the scientists were in the venture to understand that what is going on inside the plant and how is this food being made, they performed number of experiments. So these experiments which you have studied before also, but in our NCERT that is mentioned, if you just open the book, you will find that number of experiments have been performed. So the first thing, that is chlorophyll. So which experiment is going to help us to know that chlorophyll is a very important material for photosynthesis? Can you tell me? First experiment, that is the aim of the experiment was that chlorophyll is a very essential pigment for photosynthesis. So what did scientists do? They took a plant. Suppose this is a potted plant and this is a leaf from that plant, apical meristem. This is a green leaf. Obviously, I can make it green like this. Entire surface. Right. ये green plant है. Okay. And he took another plant, like for, which is showing variegated leaf. You know what is variegated leaf? Any idea? Tell me.
So they have spotted spots.
They helped. Didn't get you. Just once more. What are
they have some patches like spots.
That means variegated leaves are those leaves where the colors are, that is the pigments, that is not only it is totally green, it has different colors. That means green color is not present throughout the entire leaf, right? It is present in patches. Am I correct? So what are
Yes.
the places, that means, that other regions where this chlorophyll is not present, that is green color pigment, they are not present. There may be some other pigments like anthocyanin, like many other pigments can be present in between, like beta carotene and many more others. Like I'm making some of them in this way. So what we can call this plant as, it is a plant which shows variegated leaves. Yes, ये variegated leaves है, लेकिन ये पूरा green leaf है. Green throughout. Now what did he do? He used a black paper. ऐसे एक black paper लिया और ये जो portion है इसको cover कर दिया. That means with the help of a black paper, the scientists, they covered this portion of the leaf. So this portion of the leaf is not receiving sunlight, right?
Yes, sir.
And the entire setup was kept under sunlight, this entire setup, both this one as well as this one, for some period of time, say 2 hours, 2 to 3 hours. Like this. and after some times this leaf was plucked. ये दोनों leaf pluck किए गए, right? And they were taken for iodine test. In class 10 you have studied iodine test, you
Yes.
remember? Iodine is basically brick color, but when it reacts with starch, it changes into starch iodide, which is blue black in color. अगर starch इसमें मौजूद है तो you can understand that this portion is synthesizing starch. That means photosynthesis has taken place. And if starch is absent, glucose, they will be converted into starch by polymerization in biomolecules you are studying. So they will be, what is the difference between cellulose and starch? Biomolecules का class start हुआ ना, मतलब carbohydrate वगैरह.
Yes, yes.
Cellulose और, हाँ?
First page or second page are already done.
या मतलब सेल्यूलोज और स्टार्च के बीच अंतर क्या है? जो भी आए तुम बोलो.
Starch and cellulose, man.
Yes.
I don't know.
Mm-hmm.
No, ma'am, I don't know.
Okay, okay. See, both of them, they are polymer of glucose, but one is, in case of cellulose, they are the polymer of beta-D glucose, and they are unbranched. Only 1,4 glycosidic linkage, this she will be dealing. Only 1,4 glycosidic linkage is present in cellulose. They are unbranched polymer. They are present in form of sheet, जैसे ऐसे ऐसे present होता है. और इसके अंदर जो सेल्यूलोज का माइक्रोमोलिक्यूल है, मतलब सेल्यूलोज ऐसे होता है, और स्टार्च में क्या होता है, बीट that is alpha 1,4 glycosidic linkage is present. Along with that, there is presence of 1,6 glycosidic linkage. Alpha 1,4, this is known as amylose, and 1,6 is known as amylopectin. तो ये amylopectin, ये स्टार्च बनने के बाद ऐसे हेलिकली वो टर्न हो जाते हैं. Why I am saying, because I need to explain why the color changes. So,
Oh,
did you know this from before?
no, ma'am.
So, iodine, ये जो iodine है ना, तुम iodine use करोगे तो iodine यहाँ पे चिपक जाएगा. That means it will enter, it will act as a mordant or chelating agent. So, therefore, in presence of refractive index, that means the refractive index of this will change and it will give a blue-black color. Moreover, with amylose and amylopectin, they are giving different coloration, and the combination of that particular color also is responsible for appearance of this particular color, that is blue black. So now coming back to our own scenario, that is when this portion, this part, one leaf has been covered and everything was, the entire setup was placed under light, it was found when this leaf was taken for iodine test, it was taken for iodine test. So what it shows, that in all these portion, it turns blue black. So I'm drawing the leaf again. Here you can see that after iodine test, it looks blue black in color. So I'm just taking the blue color over here, right? Because blue black combination is not there in the color combination. Yes, blue black color is present except for the middle part. Why? Because in this region there is no formation of starch. These are the portion where starch has been formed. Understood? Starch is
Yes.
formed. That means starch is formed means there is synthesis of food material, there is synthesis of carbohydrate, and starch is formed. And when you put iodine in these regions, the entire leaf, that means you are taking it for iodine test. First of all, in iodine test, you know that you have to boil the leaf so that you can de-starch. First, you have to keep the plant for in dark for some days, overnight, and then you have to pluck the leaf. So here you are putting the leaf under sunlight. Here there is no scenario of dark because already dark paper you are putting it over here, and the leaf you are taking it. First you have to fix the leaf in the very hot water, and then you have to clear it with the alcohol, that is warm alcohol, not very hot because it will catch fire. So what will happen, the chlorophyll being that is non-polar solvent, it will get cleared out, and the starch will remain. So when on giving iodine, iodine will get into the helical structure of starch, and by change of the mu, that is the refractive index, you can find that the other parts which were not covered shows blue-black color. While the central portion, this portion where no, that means the paper was present, no coloration was found, no blue-black color was found. Understood?
Yes.
Again, on plucking this particular leaf, what has been found? It has been taken for the iodine test, and there also these regions, yes, these regions which were green in color, they show blue-black. So what was the inference from this particular experiment? This is clear to all, this is clear to you?
Yes, sir.
Has anyone other joined? So here we can, this part ये समझ में आया? पहले
Yes.
हम क्या किया, मैं ये लीफ लिया, इसको कवर किया और उसके बाद मैंने आयोडीन डाला इसके ऊपर, तो जिस पोर्शन एक्सपोज्ड है, ग्रीन है, उसके अंदर स्टार्च का फॉर्मेशन हुआ, तो वो ब्लू ब्लैक कलर शो किया. तो अंदर वाला जो जहाँ पे ये पेपर मौजूद था, इट इज नॉट शोइंग एनी कलर चेंज. On the variegated leaf, we can see only the green portions because it is a mosaic condition where different green and other color combinations due to different pigments they are present, and we can see that blue black is found only in the green portion. Clear?
Clear.
So from here, can you tell me that what can be the inference? यहाँ से हम inference क्या draw कर सकते हैं? Yes, कोई
So
inference?
from here we can take that chlorophyll is very essential for the plant and it catches the sunlight.
It is essential for photosynthesis. So chlorophyll is essential. Essential for photosynthesis. ये photosynthesis के लिए जरूरी. So why we are doing this? Because जैसे मैंने तुमको पहले पूछा, I asked you before that what are the things which are necessary for photosynthesis. तुमने क्लोरोफिल बताया, उसके साथ लाइट बताया, कार्बन डाइऑक्साइड बताया, है ना? तो पहले से साइंटिस्ट को क्या ये पता था? Did scientists know this from before that these are very essential substances? No. They performed experiment, and this is one of the experiment which establishes yes, chlorophyll, it is essential for photosynthesis. Clear हुआ?
Yes, ma'am, clear.
Can you see the board?
Yes, sir.
Now, this scientist, what did he do? Yeah. Now, in case of this mole's half-leaf experiment, what is found is that he tried to show, that means he had taken a plant in order to see that, suppose ये plant है, इसका leaf पहले से फिर से बना रहे हैं, leaves है, और ये जो एक leaf, इसको मतलब एक bottle लिया गया, that means a bottle has been taken, a portion of this leaf has been taken into that container like this. ये एक container है. You also follow it from NCERT, the experiments जो दिया हुआ है ना, तो वो मैं पहले डिस्कस कर रहा हूँ और उसके बाद मेन टॉपिक में आ रहा हूँ.
Oh,
देख रहे हो NCERT को?
yes, sir.
ये कंटेनर है और इसमें सोक्ड कॉटन रखा हुआ है. दिस इज सोक्ड कॉटन लेकिन किसमें सोक्ड है? KOH में, पोटेशियम हाइड्रोक्साइड है। तुमको शायद पता है ये जो पोटेशियम कार्बन हाइड्रोक्साइड है, ये किसको एब्जॉर्ब करते हैं? कार्बन डाइऑक्साइड को एब्जॉर्ब करते हैं, राइट? इट
Yes.
एब्जॉर्ब्स कार्बन डाइऑक्साइड। तो ये जो कंटेनर है, यहाँ पे जो गैसेस है, उसमें क्या कार्बन डाइऑक्साइड होगा? कार्बन डाइऑक्साइड नहीं मौजूद होगा। सो आई हैव पुट अ क्रॉस असाइड, राइट?
Yes, sir.
और ये पूरे सनलाइट के अंदर हो रहे हैं। तो आफ्टर समटाइम्स वी जस्ट टेक दिस लीफ, एंड ये लीफ को लेकर हम क्या करेंगे? स्टार्च टेस्ट के लिए ले जाएंगे। वी विल टेक इट फॉर स्टार्च टेस्ट। स्टार्च टेस्ट के लिए इसको ले जाएंगे, और इसके बाद क्या देखेंगे? तुम बोलो,
Sir,
कोशिश करो।
yes, sir. If we take for test, we will dip it into iodine.
हाँ हाँ।
Sir, as we have to perform the first experiment,
हम्म।
then we have to dip it into iodine.
यस, आयोडीन स्टेज, मतलब स्टार्च टेस्ट मतलब आयोडीन टेस्ट। तो आयोडीन टेस्ट जो जैसे प्रोटोकॉल है वैसे ही हम लोग फॉलो करेंगे, और इसके जो रिजल्ट होगा वो क्या हो सकता है?
Yes, sir. If we dip it into the iodine, then it will not form the bluish color.
That means entire leaf will be bluish black, or a portion of the leaf will be bluish black.
Yes, sir.
Which one? The first one or the second one?
It first one.
The entire leaf will be bluish black.
Uh,
That is,
as the half covered, the half will turn.
that means half will be blue black in color, and half will not be blue black in color. Yes,
Yes.
this one or that one.
Uh, the second one, second one.
That means this portion will be blue black, while this portion will remain as it is. So what can be the inference from your side? That means this portion, this is the green part. Yes, you know that this portion was green. The entire leaf was green. So there is no problem with chloroplast. There is no problem with chlorophyll. So when light falls, this portion, it is getting carbon dioxide. ये carbon dioxide यहाँ पे, it is getting carbon dioxide.
Yes, sir.
But this portion, it is getting light. It is green in color, but not getting one factor. So from this, we can also conclude that carbon dioxide is essential for photosynthesis. Am I clear?
Yes, sir. Got it.
So after this Mole's half-leaf experiment, why it is called half-leaf experiment? क्योंकि पोर्शन, आधा पोर्शन लीफ का लेकर ये एक्सपेरिमेंट किया गया है। तो थर्ड इज, I'm just then another experiment was performed by Joseph Priestley. SE. Now this Joseph Priestley, what did he do? He have taken a bell jar. Inside the bell jar, this is a bell jar, right? Inside the bell jar, he have taken a candle. It's a burning candle like this. After some times, he found that the candle extinguished. After some times, candle extinguish. After some time ये जो कैंडल है ये extinguish हो गया, क्योंकि इसके बाद उन्होंने क्या किया, ये तो पहले वाला चीज कर दिया, उसके बाद इसके सेम बेल जार ने लिया, the bell jar has been taken and he took a mice. It was found that the mice also dies after some time. ठीक है, लेकिन ये सेकंड वाला काम उन्होंने किया. तो थर्ड, what did he do? He have taken the bell jar, he took the candle which is burning, he took the mice also, along with that he put a mintha plant, that is a mint plant over here. This is a mint plant like this. So this is mint plant. Mint plant. So what did he see? That mice survived and the candle also went on burn. He found that when this mint plant has been introduced into the bell jar, the mice survived. Here the mice was dying. Can you see? Hello?
Yes, sir.
So mice died and the mice survived and the candle also kept on burning. So from here he came to the conclusion that the air was, that means some important thing of the air was utilized by this burning candle and respiring animal mice, and they produce a foul air, and this foul air, it is known as phlogiston. Phlogiston, right? And when this particular mint plant has been introduced, it absorbed the foul air, that is phlogiston, and convert it into dephlogiston. It converted into dephlogiston, right? So later on it was came into concept that dephlogiston is oxygen and phlogiston is carbon dioxide. So from here it is very clear that oxygen is produced in the process of photosynthesis. In this process, oxygen is also produced. Entire thing was kept under sunlight, that was obvious, and oxygen was produced in the process of photosynthesis. Clear?
Yes, sir.